This is Chapter 11, Part C, on the nervous system and nervous tissue. So the nervous system works because information is able to flow from neuron to neuron. So neurons are functionally connected to one another by synapses. So this is what's going to mediate that information transfer. So this is the site where different neurons can communicate from neuron to neuron or from neuron to an effector cell. So this is showing a synapse between two neurons. So one neuron, our presynaptic neuron, is going to release the neurotransmitter chemical messengers that would then be received on the dendrites of the receiving postsynaptic neuron. So looking at with an effector cell, like with muscle contraction, so at that neuromuscular junction, we had acetylcholine released from the axon terminals that binds to those receptors on the actual muscle cell surface. So the presynaptic neuron is the neuron that is sending the impulses toward the synapse or toward that communication point. The postsynaptic neuron is after the synapse. So it's going to receive that signal and then carry it away from the synapse. So in the peripheral nervous system, this postsynaptic receiving cell could be another neuron, or it could be one of those effector organs that we talked about. So a skeletal muscle cell or a gland. So many neurons are able to function as both. So remember we said neurons can carry on multiple conversations simultaneously. So one neuron could be sending a signal to this neuron while simultaneously receiving information from another neuron. So there's two main types of synapses or information transfer methods. So we can transfer this information via a chemical synapse or an electrical synapse. So chemical synapse is going to use neurotransmitters. So it's going to communicate with these chemical signals, whereas an electrical synapse is going to utilize gap junctions and transmit that electrical signal directly to the receiving cell. So chemical synapses are the most common type. We've talked about these before with the neuromuscular junction and muscle contraction. So these are specialized for release and reception of chemical messengers called neurotransmitters. So chemical synapses typically are composed of two parts. So you have your axon terminal of your presynaptic neuron, of our sending cell, that's going to contain synaptic vesicles or just little sacs of neurotransmitter. The receptor region is located on the postsynaptic cell membrane. So it's going to receive that neurotransmitter chemical messenger. So this is typically going to be on the dendrites or a cell body. So notice they're not actually physically connected. There is a bit of empty space between these two. So this is called the synaptic cleft. It's just a fluid-filled space where these neurotransmitters will kind of float across to their receptors. So in chemical synapses, we initially begin with that electrical impulse, right, that action potential, but we're going to convert that electrical message into a chemical message across the synapse. And then from there, it would be converted back into an electrical message. So for instance, with your neuromuscular junction, skeletal muscle contraction. So once acetylcholine binds to these receptors, it's going to kickstart the process of that action potential or generating another electrical signal. So once a neurotransmitter has done its job, we need to terminate its effect. So we don't want the neurotransmitter to continue stimulating a cell unless it's required. So as long as that neurotransmitter is binding to the receptor, those graded potentials will continue. Right, so we need to regulate this process. So within a few milliseconds, the neurotransmitter effect would be terminated in one of three ways. Right? So really that neurotransmitter is just kind of the initial messenger. So once the cell receives that message, we no longer need that neurotransmitter. So we could just do reuptake. So some of those astrocytes, those neuroglial cells that help to mop up and clean up the immediate area around neurons. We could have enzymatic degradation. So there's a specific enzyme that would work to break down these neurotransmitters. So in the case of acetylcholine with muscle contraction, the enzyme is called acetylcholine esterase, or ACE. So it's going to break down that acetylcholine neurotransmitter to stop stimulating the muscle cell. And then finally, just simple diffusion. So given enough time, the neurotransmitter will just kind of float away from that binding site. So electrical synapses are a bit less common than chemical synapses. So in these types of connections, the neurons are electrically coupled. So they're sharing their direct electrical signal from cell to cell. There's no conversion to a chemical signal back to electrical. So with these, communication is very rapid and usually unidirectional or bidirectional, meaning it only goes one or two directions. There's not a whole lot of detours or side tracks for these impulses. So these are common in cardiac muscle. So gap junctions, those intercalated discs of the cardiac muscle, are where the individual cardiac muscle cells are electrically joined together. So this prevents every individual muscle cell from having to have its own individual stimulation. So we want heart muscle to contract kind of as that single coordinated unit. So we do this with the gap junction. So they all kind of share the same impulse and respond to the same impulse at the same time. So neurotransmitters, we said, were just the chemical messengers of the nervous system, or the language of the nervous system. So there are 50 or more neurotransmitters that have so far been identified. Most neurons make 2 or more of these neurotransmitters. So one neuron can actually have different effects or influences on different cells, but they're going to be released at different frequency. So one cell might release adrenaline and serotonin, but it's only going to release adrenaline when it's stimulated for that particular neurotransmitter. So we classify neurotransmitters based on their chemical structure and their function. So again, everything in anatomy and physiology we can look at based on its structure and its function, even down to the chemical and molecular levels. So acetylcholine is the first identified and best understood neurotransmitter. So we've already been introduced to acetylcholine in skeletal muscle contraction. So we know it's released at the neuromuscular junctions, as well as some autonomic nervous system neurons and some central nervous system neurons. And like I said, it's degraded by that enzyme acetylcholine esterase. So again, showing the neuromuscular junction, the action potential or the electrical impulse signal reaches that neuromuscular junction, causes the axon terminal to release acetylcholine, which binds to specific receptors on the surface of the muscle cell, which ultimately lead to an action potential and muscle contraction. Another class of neurotransmitters are biogenic amines. So these include a couple different types. So catecholamines are things like dopamine, norepinephrine, epinephrine. So all these have in common is that they're made from the same amino acid. So looking at their chemical structure. Indolamines are things like serotonin and histamine. So histamine is one of the primary inflammatory chemicals in the body. So like if you have allergies, you take Benadryl, which is an antihistamine, which prevents some of that inflammation and irritation with allergies. So all of these are widely used throughout the brain, playing roles in emotional behaviors as well as biological clock. And some of the imbalances with these particular neurotransmitters can be associated with mental illness. So particularly serotonin. So a lot of commonly used antidepressants, anti-anxiety medications are classified as SSRIs. So these are selective serotonin reuptake inhibitors, right? So they essentially are going to block the reuptake of serotonin neurotransmitter, causing it just to hang around a bit longer in the synapse. So we get kind of a longer lasting effect of that serotonin. Epinephrine is another name for adrenaline. So when you have your adrenaline's rushing, right, your heart's racing, your respiration rates increase, that would be associated with some of that sympathetic nervous system. Your fight or flight response is going to be dictated by this epinephrine or adrenaline neurotransmitter. Amino acids can also act as neurotransmitters. So as we know, amino acids are the building blocks for proteins. So because they're already very common, it's hard to kind of pick apart which ones act as neurotransmitters. But there are a few that we have proven. So things like glutamate, aspartate, glycine, GABA. So aspartame is common artificial sweetener. So really it's just made up of this aspartic acid, this aspartate, with a phenylalanine group and a methanol. Okay? So the aspartate is part of your aspartame. So here's glycine, glutamic acid, and GABA. Peptides or neuropeptides are just strings of amino acids that can play a role as neurotransmitters. So things like substance P helps to mediate pain signals from the body. Endorphins are kind of the body's natural opiates or natural pain relief. So they just help to reduce the perception of pain. Gut-brain peptides also play a role in regulating the digestive process. So cholecystokinin is released by the duodenum, part of the small intestine, to stimulate release of other digestive enzymes from the pancreas and the gallbladder. Purines are nucleotides, so part of the building blocks of DNA and RNA. So the building blocks of nucleic acids can also have a neurotransmitter effect on the nervous system. ATP, even, our primary energy molecule, can now be considered a neurotransmitter. So the primary purine neurotransmitter is adenosine. So it acts as a pretty potent inhibitor in the brain. So caffeine works by blocking the receptors for adenosine. So if we look, adenosine and caffeine both have very similar chemical structures. So this just means that caffeine is able to fit into our receptors for adenosine in the brain, in the nervous system. So essentially blocking the adenosine from binding. So adenosine, you can think of kind of like your sleepy chemical. So once we have enough adenosine build up in the brain and the body, right, all of our receptors get filled, then we have that tired, sleepy feeling. But caffeine temporarily blocks those receptors. So we may still be building up lots of adenosine, lots of this sleepy chemical, but it's not able to be detected by our cells. So we don't feel that sleepiness until the caffeine wears off, and then it opens up these binding sites, and now all of the adenosine is going to rush in and fill all of the sites, and that's when you get kind of that crash after a caffeine high. Some gases and lipids can act as gasotransmitters, neurotransmitters. So things like nitric oxide, carbon monoxide, hydrogen sulfide. So they work by binding with some of those G protein coupled receptors in the brain. So because they're lipid soluble, they're able to be synthesized more readily on demand. So nitric oxide is a common vasodilator given to patients. So here showing a constricted blood vessel. So if we need to open up that blood vessel, increase the blood flow, the cells can release nitric oxide, causing the smooth muscle in the blood vessel to relax a bit, to vasodilate. So now we open up those blood vessels and increase the blood flow to a particular area. Endocannabinoids are naturally occurring neurotransmitters that are chemically similar to THC in marijuana. So they're going to act at the same receptors as that THC. So these cannabinoids, endocannabinoids, are lipid soluble, believed to be involved in learning and memory, as well as neuronal development, controlling appetite, suppressing nausea. So this is why medicinal marijuana is good to give to patients with like undergoing chemotherapy or AIDS, because some side effects of those diseases and treatments could be loss of appetite, increased nausea. So the THC fits in those same receptors as these endocannabinoids in the brain. So now we can look at classification of neurotransmitters based on their functions. So we know neurotransmitters have a great diversity of functions, but we can split it up into two main classifications. So we can look at the effects of a neurotransmitter, or we can look at their actions. So effects could be either excitatory or inhibitory. So an excitatory synapse is going to stimulate depolarization. So we're going to excite that cell to generate an action potential or an impulse and do something. As opposed to an inhibitory synapse, we're going to essentially cause a hyperpolarization. So we're going to turn off that cell and prevent it from generating action potentials or responses. So the effect of the neurotransmitter is really going to depend on the receptor it's binding to. So acetylcholine and norepinephrine bind to at least two different types of receptors that have opposite effects. So we know acetylcholine is excitatory and stimulates skeletal muscle, right, at that neuromuscular junction. Right, so that was kind of our kickstart signal for muscle contraction, was acetylcholine. However, if acetylcholine is released to cardiac muscle, it would have an inhibitory effect. So instead of stimulating the muscle contraction, it would inhibit the strength of the muscle contraction in the cardiac muscle. So it all really depends on kind of our final receptor and what our end goal is. So actions could be direct versus indirect. So a direct action is when the neurotransmitter binds directly to the channel and opens it. So this would be a much more rapid response to generating those action potentials. So acetylcholine was a good example that we've already looked at. So acetylcholine binds directly to those receptors on the muscle cell membrane, causing the channels to open and sodium to flow in. So an indirect action would mean the neurotransmitter has to act through this intracellular second messenger system. So the neurotransmitter doesn't elicit the direct effect on its own. It requires some assistance to kind of carry that message down the membrane for it. So these indirect actions generally have longer lasting effects that are more similar to hormones, whereas your direct action, once the neurotransmitter unbinds from the protein receptor, then it's going to stop the effect. It's going to close the channel. So neural integration is looking at neurons functioning together in groups. So far we've really only talked about kind of an individual neuron and its structure and function, but we know that neurons don't act solely independently. They're always interacting with other neurons and carrying on multiple conversations simultaneously. So these groups of neurons and working together are what contribute to our broader, higher mental functions. So there are billions of neurons in the central nervous system. So we have to have some type of integration between these neurons. They have to be working together some way so that the individual parts can make a smoothly operating whole. Right, so we have, for instance, our sensory receptors, those processing centers in the brain. We have our motor pathways, right, with your somatic system, with your skeletal muscles. We also have motor pathways for your autonomic system, like with your smooth muscle, your cardiac muscle. And then we also have those higher mental functions, so things like memory, learning, intelligence, all ties back into this neural integration. So looking at how these neurons are organized. So they're organized in what's called a neuronal pool. So we have functional groups of neurons. So groups of neurons kind of working together for a common shared goal. So they integrate incoming information perceived from the receptors or other groups of neurons, other neuronal pools. And then they forward that information on to its final destination or wherever it needs to go from there. Right, so we'll look at these different patterns of neuronal pools. Right? So things like divergent, convergent, serial, parallel, or reverberating. So a very simple neuronal pool would be where you have a single presynaptic fiber. So you have one neuron that's delivering the message, but then it's going to branch out to several other neurons in the pool and deliver that same message to multiple neurons at once. So the discharge zone are the neurons that are closer to that incoming fiber. And your facilitated zones are the ones kind of off to the side on the peripheral sides. So these aren't as likely to get as strong of a stimulation. So the closer the neuron is to that presynaptic incoming fiber, the stronger the signal and message will be received. In serial processing, we have input travel along just one pathway to a specific destination. Right? So one neuron stimulates the next one, which stimulates the next one. So this is a very kind of expected pattern of information transfer. So basically from point A to point B to point C. There's no kind of diverging, no shortcuts or sidetracks with this. So these generally work in your all or none manners to produce a specific but anticipated or expected response. So the best example of your serial processing would be your spinal reflexes. So things that every time that reflex is stimulated is going to give the same exact response. So like the knee-jerk reaction when you go to the doctor and they tap your knee with a hammer to make your leg kick. So that's a spinal reflex. So every time that the doctor hits your leg in the right spot, it's going to have that same predictable reflex response due to the serial processing. So showing reflexes with the serial processing. So again, these are rapid automatic responses to stimuli. So a particular stimuli will always cause the same response or reaction. So these are called reflex arcs. So 5 basic components to a reflex arc. You have to have a receptor, so some type of sensory receptor. So in the example of the knee jerk test, the receptor would be the stretch receptors in the tendons and the muscles in the leg. So receptor sensory neuron is then going to deliver that information to the central nervous system where it will be processed by the integration center, right, our interneuron. So then once we process the information, we determine an appropriate response, how we're going to respond to this information, this stimulus. We send that message back to the effector organ via a motor neuron. So again, effector organs could be skeletal muscles or glands. So in parallel processing, we have input that travels along several pathways. So different parts can deal simultaneously with the information. So you could have one stimulus, but different possible responses. So it's different with serial processing. So serial processing, we get the same response every time. Parallel processing, we may come up with different responses to the same stimuli. So this is important in your higher level mental functioning. So classic example, smell. So smell is one of the strongest triggers of memory. So one particular smell, the same smell, could remind you of any associated experience. So it could remind you of, so if you smell some food, right, it could remind you of your grandma's cooking when you were growing up, or it could remind you and make you think that, oh yeah, I haven't had lunch today, I'm hungry, right? So one stimuli can give different responses. So circuits are patterns of the synaptic connections in your neuronal pools. So there's 4 basic types of circuits that we will look at. So diverging, right, where it's kind of branching out farther from the source. Converging, where we have multiple sources kind of uniting on one focal point. Reverberating, right, where it kind of echoes or loops back around. And then parallel after discharge. So a diverging circuit, we have one input but many outputs. So it's diverging, it's branching out and spreading wider and wider. So this is sometimes referred to as an amplifying circuit. So an example would be a single neuron in the brain can then go on to activate 100 or more neurons in the spinal cord or skeletal muscle fibers. A converging circuit is when we have multiple inputs, but they're all going to focus and centralize on one concentrating circuit or one output. So again, the example of kind of that parallel processing. So different stimuli can elicit the same memory. Right? So for instance, if you have right, different stimuli, different things can make you come to the same conclusion. A reverberating circuit, the signal travels through a chain or a series of neurons, but then each one is going to kind of loop back or feedback to the previous neuron. So this is sometimes called an oscillating circuit. So this is more for controlling your rhythmic activity. So breathing, sleep-wake cycle, repetitive activities like walking. Right? So we send the message and then we kind of double back and double check. So a reverberating or an oscillating circuit. And then finally, parallel after discharge is when the signal stimulates the neurons that are arranged in these parallel arrays or arrangements, but eventually they're then going to converge on a single output. So the impulses reach this output cell at different times. So fastest point from point A to point B is a straight line. So this impulse would get here first, followed by this one, and then this one would probably get here last. So this just causes kind of a burst of impulses. So we go boom, 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 instead of all arriving at the same time. So this is where we get the after discharge. So this is involved in more of your, again, higher level mental processes like mathematical calculations.